Okay, class. Assalamualaikum. So basically, for today's lecture, so hopefully we are going to cover the whole of this chapter. Okay, up to the end. In within these 2 hours. Okay, hopefully earlier. Okay. So basically, for today's lecture, we are going to cover on antibody-mediated immune response. Tapi saya tengok ni skrin saya tak bergerak. Kejap ya. Tak bergerak kan skrin saya? Bergerak ke tak?
Dia freeze, madam.
Kejap. Okay. Alright, so basically last lecture kita tengok peranan macrophage, dendritic cell and also B cell, okay, in activating the humoral immune response or antibody-mediated immune response and also cell-mediated immune response. So saya harap by now you should be able to know that in the third line defense ada dua immune response yang kena diaktifkan. Satu humoral immune response ataupun kita panggil dia juga antibodies mediated immune response, dan lagi satu adalah cell mediated immune response. So each one of these immune response ni dia melibatkan dua jenis lymphocyte. Kalau yang antibody tu dia melibatkan B cell lah. Kalau yang cell mediated immune response dia melibatkan cytotoxic T cell. Tapi in order to activate these two immune response, kena macrophage ataupun dendritic cell ataupun B cell engulf bakteria ataupun pathogen tu lah. So bila dia engulf, dia akan fragmentize the pathogen, and then macrophage dendritic ataupun B cell ni akan menghasilkan sejenis molekul, kita namakan dia sebagai class 2 MHC molecule kan. MHC dia stand for major histocompatibility complex. Okay, so bila ketiga-tiga sel ni dah menghasilkan class 2 MHC molecule, so this molecule akan bind kepada antigen fragments, pathogen yang dah dihancurkan dalam macrophage tu tadi. So dia akan bind kepada MHC tadi. So dia akan jadi antigen MHC 2 complex. Okay, so dia akan pergi kepada cell surface. So sekarang ni bila dia dah present dekat cell surface, ketiga-tiga sel ni dikenali sebagai antigen presenting cell, APC. Okay, so dia akan present kepada siapa? Dia akan present kepada helper T cell. Okay, helper T cell. So T cell ni ada dua jenis. Satu helper T cell, satu lagi adalah cytotoxic T cell. So untuk interact dengan APC tu mesti adalah antara interaction antara APC dan juga helper T cell. So bila ada interaction ni, helper T cell ni dia ada receptor kan dekat dia punya cell surface. So receptor tu akan bind kepada antigen that is being presented by the APC. Okay, so bila receptor pada helper T cell ni bind kepada antigen that is presented by the APC, this causes the APC to release signaling molecule, we call it as cytokine. Okay, cytokine samalah asalkan ejaan awak tu betul. Okay, cytokine. So APC ni bila dah secrete cytokine, so cytokine daripada APC ni akan aktifkan helper. Okay, so helper ni bila dia dah bila dah dia terima cytokine daripada APC ni, dia pula akan menghasilkan cytokine. Okay, so cytokine daripada helper T cell ni akan ada 3 peranan dia. Satu, aktifkan lagi helper T cell ni so that the helper T cell will proliferate, divide into many cells, many helper T cell with the same receptor for the antigen. Dan bila dah terhasil banyak cells of the helper T cell, we call it as dia akan undergo clonal selection lah kan. Clonal selection akan jadi effector dan juga memory. Itu untuk helper. Dan cytokine daripada helper T cell ni dia guna juga untuk aktifkan B cell, okay, B cell that has receptor that matches with the antigen tu tadi, okay, dan juga dia akan aktifkan cytotoxic T cell, okay, that has receptor that matches with the receptor being presented by the APC tu tadi. So kalau dia aktifkan B cell, so maksudnya dia melibatkan antibody-mediated immune response lah, humoral immune response. So B cell ni dia akan bila dia dah diaktifkan, so dia akan menghasilkan effector dan juga memory. Dia undergo clonal selection lah. So lepas tu bila dia undergo clonal selection, yang effector B cell tu dia akan dikenali sebagai plasma cell. Okay, so plasma cell ni yang akan secrete antibody. So this antibody secreted by the plasma cell ataupun effector B cell tu, dia mempunyai antigen binding site that complement with the antigen that was presented by the macrophage or APC tu tadi. Okay, so kalau untuk cell mediated immune response, dia akan musnahkan, it destroys the cytotoxic T cell that was activated. So dia akan sama juga undergo clonal selection, hasilkan effector, hasilkan memory. So kalau effector yang akan aktif pada masa you are infected tu, okay, so dia akan cari infected cell, okay, infected cell that is infected by that specific pathogen that was recognized by the APC tu tadi. So cytotoxic T cell ni dia akan bind kepada MHC molecule of class 1, okay, being presented by infected body cell. Infected body cell ni dia akan menghasilkan protein dia akan menghasilkan protein dalam sel tu, kita panggil dia juga MHC, okay, MHC class 1. Kita semalam hari tu kita tengok kan perbezaan antara class 1 dengan class 2 kan. So class 1 oleh nucleated cell. Okay, so in this case infected body cell, obviously that has nucleus, will present the antigen fragment. Okay, so this is the infected cell. So dia akan infected cell akan present antigen fragment using MHC class 1. Okay, so dia akan presentkan kepada siapa? Kepada cytotoxic T cell. Okay, so this cytotoxic cell T cell bila dia dah bind kepada antigen being presented by the infected cell, so cytotoxic T cell ni dia akan musnahkanlah infected body cells tu menggunakan 2 jenis chemical, iaitu perforin dan juga granzyme. Okay, perforin dan juga granzyme. Sama juga macam kita, ya.
Macam freeze juga lah madam.
Tak, memang saya bercakap je ni. Saya bercerita je.
Okay, sorry madam.
Saya bercerita. So awak kena menghayati cerita saya ni lah. Okay, so saya cerita tanpa menggunakan apa-apa slide ni. So dia punya mekanisme dia post by the cytotoxic T cell ni sama juga dengan NK, tapi NK natural killer cell dia terlibat dalam innate immunity kan, second line defense. Ini kita cerita pasal third line defense. Okay, tapi mekanisme dia sama, dia akan secrete 2 jenis protein, which is perforin and also granzyme. Okay, so perforin ni that is released by the cytotoxic T cell ni will cause pores to be formed on the plasma membrane of the infected cell. So once pores is produced, okay, on the plasma membrane of infected cell ni, so next, okay, cytotoxic T cell ni akan menghasilkan granzyme pula, kan? So granzyme akan masuk ke dalam infected cell tu melalui dua cara. Kalau ada dalam buku, dia tulis satu by endocytosis, okay, endocytosis. Satu lagi adalah melalui pore yang terhasil tu tadilah, okay. So dia akan masuk ke dalam infected cell tu. So granzyme ni akan induce apoptosis, okay. Apoptosis which is program cell death lah ataupun cell suicide lah. So maksudnya selnya akan musnah dengan menghasilkan macam tiny vesicle macam tu, okay. So dia akan musnah. So once this infected body cell has been destroyed, so dia akan exposekan pathogen dalam sel tu tadi. Dia akan exposekan kepada siapa? Kepada satu phagocyte, which is contohnya macrophage. So macrophage bila dia terjumpa yang expose pathogen tu, dia akan datang engulf. Satu lagi mekanisme adalah bila macrophage tu dah di-expose, so akan datang antibody. Okay, so antibody adalah antibody yang dihasilkan oleh plasma sel dalam antibody-mediated immune response ataupun humoral immune response. So sebenarnya proses immune system ni dia saling memerlukan lah, dia melengkapi lah. Maksudnya kena ada dua-dua, humoral immune response dan juga cell-mediated immune response tu lah. Okay. So kalau nanti kita akan tengok kalau antibody ni cara dia attack pathogen ataupun antigen adalah satu macam tadilah, bila antibody dia circulate in your body fluid, bila dia jumpa that specific pathogen, dia mesti cari specific pathogen pathogen that was engulfed by the APC tu tadi itulah. Yes, specific pathogen, sebab dia hanya akan antibody ni dia akan ada antigen binding site kan. So antigen binding site ni will only bind to that specific antigen of the pathogen that was recognized by the APC, okay, by the APC. So antibody ni bila dia bind kepada pathogen, that specific pathogen, salah satu cara dia akan terlibat dalam memusnahkan pathogen ni adalah melalui neutralization. Okay, so dia ada beberapa cara, nanti kita akan tengok neutralization salah satu dia. So maksudnya bila antibody bind, so virus ni dia tak boleh nak bergerak lah, macam dia kena gari lah macam tu kan. Okay, so lagi satu adalah agglutination. Okay, dia akan menyebabkan dissolve antigen to agglutinate. Tak, bukan dissolve, antigen to agglutinate. Okay, dia clump together. And then lagi satu adalah precipitation. Precipitation adalah untuk antigen yang boleh dissolve. Okay, so bila dissolve, bila antibody bind, so dia akan menyebabkan precipitation, okay, of the antigen. So bila dah berlaku sama ada neutralization, precipitation ataupun agglutination, so nanti macrophage datang, dia engulf, dia destroykan. Macam tu. Okay, so habislah dah. Sebenarnya basically pasal antibody-mediated immune response ataupun cell-mediated immune response secara keseluruhannya tanpa kita tengok kepada slide. So kalau siapa yang dah baca, dah tengok video dalam acid biology tu, so then awak bolehlah imagine apa yang saya ceritakan ni. Okay. Okay, so tak apa, nanti awak tengoklah balik recording tu bila dah dah habis khatam immune ni sampai habis notes ni, so akan faham macam mana immune response ni berlaku. Okay. Okay, so kita tengok the slide. Okay, nah, daripada saya pergi cerita tadi, so this is the slide. So sekarang ni kita nak tengok antibody-mediated immunity ataupun humoral immune response. Okay, kalau humoral immune response dia daripada buku Campbell, antibody-mediated immunity adalah daripada buku Solomon. So both refer to the same thing. Okay. So it says here, for point A, APC takes in bacterium. So APC lah yang sama ada macrophage ataupun dendritic cell ataupun B cell. So this one here. So APC takes in bacterium, breaks it down and present foreign antigen on its surface in combination with MHC class 2 antigens. Okay, so nampak? So dia engulf, so nanti dia akan fragmentize kan dan dia akan present on its cell surface ni. So dia akan jadi antigen MHC complex. So nampak bakterium ni kan dekat cell surface dia contohnya ada antigen. So bila dia dah masuk dia akan dimusnahkan. So antigen fragment akan bind kepada MHC class 2. Ingat ya, class 2 by the macrophage. Okay, so dia akan present kat cell surface, okay, using class 2. So this one we call it as antigen MHC complex. So point 1B, so B cell independently combines with foreign antigen on surface of bacterium, but B cell is typically not active until stimulated by activated helper T cell. TH ni adalah helper T cell ya. So maksudnya kita dah tengok kan kalau B cell ni apart from it involved in the humoral immune response, dia juga boleh menjadi APC. Okay, dia boleh juga menjadi APC. So maksudnya tengok dekat B cell ni cell surface dia ada receptor kan. So bila receptor of the B cell binds to the antigen, so the B cell will internalize or engulf the bacterium. Okay, so dia akan present on its cell surface lah. Okay, tapi pada masa ni dia tak boleh diaktifkan lagi. Okay, dia tak boleh diaktifkan lagi sebab untuk mengaktifkan B cell ni adalah daripada helper T cell. Okay, daripada helper T cell. So helper T cell ni has to have interaction with the APC ni tadi. Okay, with APC. So APC will present antigen fragment, okay, using MHC. Okay, so dia akan attract helper T cell with receptor that complement to the antigen being presented. Okay, so ini adalah helper T cell. Okay, helper T cell yang ada receptor yang receptor dia match with the antigen being presented by the APC. So bila ada interaction ni, APC akan secrete cytokine. Okay, so number 2. Then number 2, mana point yang nombor 2? Nombor 2 hilang. I can't see the number 2, tapi tak apa, inilah kot. Okay, so the helper T cell are activated when their receptor combine with the foreign antigen MHC antigen complex on the APC. Inilah dia nombor 2 ni. Okay, so ada interaction antara helper T cell dengan juga APC. Okay, melalui the presentation of antigen using MHC molecule. Okay, so signaling takes place via cytokine secreted by both. Okay, by both cell, by the APC and also by the helper T cell. Okay, kedua-dua sel ni akan menghasilkan cytokine. So APC hasilkan cytokine, aktifkan helper T cell. Helper T cell akan juga hasilkan cytokine. Okay. So activated helper T cell, so bila dah cytokine daripada APC, aktifkan helper T cell. So activated helper T cell recognize antigen MHC complex on B cell ni. Okay, so B cell ni lah kan, so B cell. So the helper T cell secrete cytokines that activate B cell that has interacted with the foreign antigen. So ada interaction kat sini kan. So B cell ada receptor bind kepada pathogen tu. Okay, so B cell ni tak aktif lagi. So in order to activate the B cell has to receive cytokine released by the helper T cell. Okay, so helper T cell akan secrete cytokine, aktifkan B cell. Okay, so helper T cell secrete cytokines that activate B cell that has interacted with the foreign antigen. So B cell then divides. Okay, divides ataupun proliferates. Okay, proses proliferation tu tadi. Okay, and subsequent division produce clone of identical B cell. So this one kita panggil dia apa yang divide ini adalah proliferation. Okay, produce clone of identical B cell, we call it as clonal selection. Okay, clonal selection. You have so many B cell with different receptor. Only B cell that has receptor that match with the pathogen will be activated. That's why kita panggil dia sebagai clonal selection. They select hanya B cell that has receptor that match with the antigen of the pathogen. Okay. So ingat ya, proliferate, divide into many cells, okay, of the same receptor, and then clonal selection. So once that specific B cell has proliferate, so they akan undergo clonal selection and produce two clones, okay, which is the memory and also the effector. In this case, kalau B cell, effector adalah plasma cell, okay, plasma cell. Panggil dia sebagai plasma cell. Okay, so B cell, so tadi dia dah proliferate kan into many copies. So B cell differentiate and become plasma cells and memory cells. Awak take note dekat nota awak ni, plasma cells adalah effector B cell. Okay, effector B cell. So effector will pose its action during time of the infection. Okay, so it will produce and secrete antibody. Okay, so plasma cell secretes antibody via lymph and blood to the infected regions. Okay, to the infected region. So plasma cell ni akan secrete antibody, and the antibody will circulate in your body fluid, which is your lymph, your blood, and it can also enter into your interstitial fluids. Okay, so that's why we call it as the humoral immune response. Humoral, it refers to your body fluid. Okay, so the antibodies are specific to the antigen that activate the original B cell. Okay, so the antibodies are specific to antigen that activate the original B cell. So memory B cell will be stored, okay, will be stored in the lymph tissue after the infection has been overcome. Okay, so maksudnya memory B cell ni will be stored in the lymph tissues such as in the lymph nodes. Okay, so memory B cell will have longer lifespan, okay, because the function of the memory B cell is to be used during secondary immune response. When you are infected again by the same virus, for example. Okay. Okay, so kita tengok gambar ni, nampak banyak je macam tu kan. Tapi again, analyze diagram ni satu per satu. Okay, satu per satu. Jangan tengok sekaligus, tak tahu pening kan. So tengok satu per satu lah. So yang A ni, okay, nampak. So this is APC. So jot down kat sini, APC boleh jadi daripada macrophage, boleh jadi daripada dendritic cell, boleh jadi daripada B cell. Okay, so dia akan engulf, presentkan antigen fragment at the cell surface using MHC class 2. Okay, ingat class 2. So dia akan present kepada helper T cell. TH ni, small subscript H ni adalah helper T cell. So helper T cell with TCR, helper T cell receptor, okay, T cell receptor. That complements with the antigen being presented. So this helper T cell will be activated. Activated by how? By the cytokines released by the APC. Okay, so APC release cytokines, activekan helper T cell. So now helper T cell also secrete cytokines. Okay, so the cytokine released by the helper T cell will activate itself. The helper T cell undergo clonal selection, kan, proliferate, undergo clonal selection. So dan lagi salah satu fungsi cytokines released by the helper T cell is to activate B cell. So B cell ni initially dia dah bind kepada bacterium, okay, or the pathogen. Tapi B cell ni is not activated yet. Okay, so in order to activekan B cell ni, it requires the cytokines, okay, released by the helper T cell. So once this cytokine, okay, has activated the B cell. So now B cell will undergo proliferation. Okay, nampak dia akan divide into many cells. Okay, many cells, proliferation, and also clonal selection. Okay, clonal selection. So dia akan jadi dua clone, which is the effector. So this one, on this side you have the effector. Okay, on this side you have the memory. So kalau kita tengok point yang nombor 4, activated B cell increases in size and divide by mitosis. So dia akan divide. Okay, divide. So akan ada yang jadi effector, akan ada jadi memory. So ini akan jadi memory. So this one we call it as clonal expansion. Clone of competent B cell is produced. Clonal expansion. So ini daripada buku Solomon lah. Dalam buku Solomon ni saya tengok dia guna istilah clonal expansion. Tapi kalau dalam buku Campbell dia guna istilah clonal selection. Sama je lah. So it produces clones. Clone of effector, clone of memory. Okay. So B cell differentiate into plasma cell and memory cell. So plasma cell is the effector B cell that secretes antibody. So nampak secrete antibody. So the antibody will circulate in your body fluid to find that specific pathogen and it binds to that specific pathogen. So remember this is third line immunity kan. So dia akan bind kepada specific pathogen only that was recognized and engulfed by the APC molecule initially here. Okay. So plasma cells secrete specific antibody which are transported via lymph and blood to the infected region. So number 8, antibodies combine with the antigen on pathogen surface forming antigen-antibody complex. Okay, so this is antigen-antibody complex. So pathogen will be destroyed. So cara the destruction of the pathogen by the antibody tadi saya mention neutralization, precipitation, agglutination. Okay, ada lagi satu. Nanti kita tengok. Okay. Okay, so nampak. So bila kita go diagram ni one step by step, so dia tak ada pun susah kan. Tapi bila sekaligus awak pandang ni, awak penting je kan. So tengok satu persatu diagram ni. Okay. Dia tak susah pun. Okay, so this is a video. So I guess I need to play it. Sekejap ya. See video. Group man pula. Mana tadi media. Okay.
In its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully humoral immunity, carried out mainly by B cells, deals with invaders in body fluids. B cells are lymphocytes that send out antibody molecules in response to certain stimuli. There are many kinds of B cells. Each kind produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully binds to an antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly to future exposures to the same antigen. They account for the speed and strength of the secondary immune response that gives us long-term immunity to a disease. Plasma cells multiply rapidly. They produce and secrete antibodies in increasing amounts, peaking about 2 weeks after the activation of the original B cell. These antibodies flow through the body's fluids and tag the original foreign antigen molecules, marking them for destruction.
Okay, so that's a video to explain on the humoral immune response. So, sekejap ya. Okay, so now we are going to look at the flow lah. Okay, so antibody-mediated immunity or humoral immune response as a summary. So the first one, it says here, a pathogen invades the body. Okay, a pathogen invades the body. So next, an APC phagocytose the pathogen. Phagocytose or engulf lah. It engulf the pathogen. So APC can be macrophage, dendritic. Dendritic tu kalau dekat epithelial lining of skin, okay. And then and also B cell, okay. So next, the foreign antigen MHC complex is displayed on the APC surface. Tolong note kat sini, class 2 ya, class 2 sebab dia melibatkan APC. Okay, so next, helper T cell binds to the foreign antigen MHC complex. So only helper T cell that has receptor that complements to the antigen being presented by the APC will bind to the antigen. Okay, being presented by the APC. So next, okay, activated helper T cell interact with a B cells that displays the same antigen. So in B cell will also bind to that pathogen, okay, tapi it is not yet activated. So in order to activate the B cell, it requires the cytokine produced by the helper T cell. Okay. So now the helper T cell that secretes the cytokine will activate the B cell. Okay, so the B cell is activated. Then clone of B cell is produced. Okay, so B cell differentiate. Okay, differentiate becoming plasma cell and also memory B cell. So awak take note lah kat sini, ini adalah clonal selection ataupun tadi dalam illustration tu dipanggil clonal expansion. Okay. So dia hasilkan dua clone, plasma cell and also memory B cell. So plasma cell secretes the antibody into your body fluid, your blood, your lymph, okay, and it also can get into your interstitial fluid. And then antibody form complexes with the pathogen. So pathogen is destroyed, okay. So nanti kita tengok, okay, so now we are going to look at how does the antibody destroys the antigen. So antibodies do not kill pathogen. Dia tak boleh nak kill sebab dia tak ada ability to engulf. It is just a protein molecule, okay. So dia bukan white blood cells, okay. So antibody do not kill the pathogen. Instead, they mark pathogens for the inactivation or destruction, okay. Inactivation yang saya cakap tadi lah, proses tu melibatkan neutralization, okay. Destruction, bila dia neutralize, then macrophage will come and then destroy that pathogen, it engulf that pathogen, okay. So for neutralization first kita tengok. So in neutralization, antibodies bind to the viral surface protein, okay, antigen tu tadi lah, okay, preventing infection of a host cell, okay. So dalam awak punya badan ni, dalam awak punya darah contohnya akan ada virus tu, that specific virus, okay. So virus tu obviously on its cell surface akan ada antigen, okay, antigen that was recognized and engulfed by the APC tu tadi, okay. So this virus, okay, with antigen on its cell surface, okay, so this antigen saya draw lah. Okay, virus. Kan? So virus. Okay, so kat cell surface ni akan ada antigen. So this antigen, okay, ni dalam punya blood. Alright. So this virus dia happily circulating in your blood. Tapi tiba-tiba dia jumpa dengan antibody. Okay, so this antibody will bind to the antigen on that virus. Okay, on that specific virus. Okay, that virus, this virus, mestilah ada adik-beradik dia yang lain kan, that was initially engulfed by that by the APC. Okay, so antibody will circulate in your blood to find that specific virus and it binds to the antigen of that virus. Okay, so antibody bila dia bind, dia akan menyebabkan neutralization. Macam virus tu tak boleh nak reproduce lah kan, dia tak boleh nak reproduce, dia tak boleh nak bergerak. Faham? So dia neutralize macam tu. Okay. So and then antibody kalau dia dah tak boleh nak bergerak dah, obviously kan dia dah macam dah digari. So this prevents the virus from going into the host cell, the host body cells. Okay, so kalau dia dalam punya darah, so dengan adanya interaction macam ni, neutralization, so this prevent the virus from getting and infecting your body cells. Okay. So ataupun antibodies may also bind to the toxin in body fluid and prevent them from entering body cells. So this toxin, for example, is secreted by the pathogen. So pathogen releases toxin. So this toxin, obviously dia adalah molecule kan. So when it is a molecule, obviously it has specific shape. Okay. So this toxin will be triggered ataupun antibody will bind to this toxin molecule as well. Okay, toxin that was produced by that specific virus. So toxin in your body fluid will bind to the antibody. Okay, so this interaction, the binding of the toxin with the antibody macam tu prevents, okay, the toxin from entering your body cell. Okay, ataupun host cell. Okay. Faham Jangan takut dengan chapter ni, dia senang je. Okay. So neutralization. So antibody, so this is the virus. Okay, virus. So on its cell surface akan ada antigen. So this is antigen. So kita baca hari tu istilah apa? Epitope kan. So specific structure, okay, on the antigen of the pathogen. So setiap antigen ni dia akan ada several shape of epitope. Okay, so this apa dia punya hikmah dia supaya lebih banyak antibody with different binding site, okay, with different binding site can bind to the virus. Bukan sejenis, bukan satu jenis je antibody boleh bind kepada virus tu. So one virus will trigger the activation of several B cell, okay, that has receptor that can bind to the different shape of epitope of the virus of the pathogen contohnya. Okay, itu dia punya hikmah dia. Bukan ada virus satu dia akan menyebabkan activation of B cell 1 saja, tak. Dia boleh menyebabkan ataupun trigger activation of several other B cell. B cell 2, contohnya B cell 3, B cell 4 contohnya. Kan sebab apa? Sebab dekat virus tu ada banyak the several shape of epitope that the B cell receptor can recognize. So once all of these cells are activated, so it produces antibodies with different binding site that can bind to the virus. Okay, so satu virus boleh menghasilkan banyak jenis antibody. Sebab apa? Dia akan diaktifkan oleh dia akan mengaktifkan several B cells. Okay, several different B cells. Okay. So lagi tinggilah chance virus tu di-attack oleh your immune system. Macam tu. Okay. Okay, so tadi neutralization. Okay, so now we are going to look at this term, opsonization. Okay, opsonization. So in opsonization, antibodies bind to the antigen on the bacteria. Kan? So bacteria tu maksudnya pathogen lah, can be bacteria, it can be virus, okay, whatever that is foreign that enters into your body. Okay, so present a readily recognized structure for macrophage and neutrophil, and therefore triggering phagocytosis. Okay, so that is opsonization. When antibody binds to the bacteria, it can cause, for example, clumping. Okay, clumping of the antigen, or precipitating the antigen. So bila antigen dia dah clump together ataupun precipitate ni untuk antigen yang dissolve. Kalau antigen ni dissolve, okay, dissolve in your body fluid, so it is harder for the macrophage to come and then engulf. So the dissolved antigen has to precipitate first. Okay, bila dah berlaku precipitation ni, then only macrophage can come ataupun other phagocyte can come, such as neutrophil, and engulf that antigen that has undergo precipitation tu tadi, ataupun agglutination. Okay, so basically opsonization ni dia terbagi kepada dua. Note kat sini precipitation and also agglutination. Precipitation untuk dissolve antigen. Okay, so ataupun lagi satu yang ketiga ni, tadi ini yang pertama. Okay, cara pertama antibody destroy, okay, the antigen. Ini cara yang kedua, which is opsonization. Okay, cara yang ketiga, opsonization ingat ada precipitation and also agglutination. So cara yang ketiga, antigen antibody complexes may bind to a complement protein which triggers a cascade of complement protein activation. Okay, so ultimately a membrane attack complex forms a pore in the membrane of the foreign cell, leading to lysis. Ini kalau kita tengok mekanisme dia sama macam lebih kurang dalam innate immunity yang complement system tu kan. So dalam badan awak akan ada complement system, complement protein. So this complement protein akan bind kepada sel, so dia akan masuk kan, dia akan masuk dalam plasma membrane of the virus and it form pores. Dia akan menyebabkan pores terhasil. Okay, contoh, jap kita tengok Kita tengok opsonization dulu. So ini nanti kita pergi kepada complement protein ni. Opsonization. So nampak So this is the bacterium. So it says here. So okay, so you have the bacterium or the virus. Okay, so antibody bind to the bacterium and causes the bacteria to be agglutinated or undergo precipitation. Okay, so bila dah clump together, obviously dia tak boleh nak bebas berkeliaran dalam punya badan, faham tak? So now next macrophage will come and then engulf that bacterium, okay, that was bound by the antibody. Macam tu. Okay. Kalau tengok dalam nota ni, yang kat sini ada animation, okay. Kalau yang ni nanti saya play, yang ni adalah opsonization, okay. Opsonization. Okay. Ini. Okay. So activation of next yang tadi mechanism yang ketiga, activation of complement system and pore formation. So kat sini kita ada plasma membrane of the pathogen. Okay, plasma membrane of the pathogen. So plasma membrane of the pathogen, dekat cell surface ni akan ada obviously antigen lah kan. So antigen. Ini antigen. Okay, antigen ni adalah epitope. So bila antibody Kan, bila antibody bind kepada antigen ni, kan, antibody bind kepada antigen of the pathogen, akan datang kemudiannya complement proteins. Complement protein adalah host punya host punya, badan awak punya. Okay. So complement protein, bila dia jumpa ada interaction antara antibody dengan antigen of the pathogen, dia akan diaktifkan. So once complement protein is activated, so dia akan trigger these proteins, okay, to be embedded in the plasma membrane of the pathogen to form pores. Okay, to form pores. So this pore, the function of this pore is so that, for example, water, ions, okay, enter into this pore and into the pathogen and causes the pathogen to expand and then burst, it lies. Okay, so this we call it as the membrane attack complex. Okay, membrane attack complex yang ni forms pores in the foreign cells membrane, so the cell swell, okay, and then burst, lies, burst samalah. Okay, so nampak dia dah tulis kat sini. Okay, flow of water and ions enters into the cell through the pores formed by the complement protein due to the interaction of the antibody with the antigen of the pathogen. Okay, so itu cara yang ketiga. So basically cara antibody musnahkan pathogen adalah dia tak directly, okay, the antibody will not directly destroy that pathogen. Dia akan lead to the pathogen to be destroyed. Cara dia satu, neutralization. Okay, dia bind, so virus tu tak boleh nak bergerak dah, dia tak boleh nak infect your body cell. So next after that, macrophage come and then engulf. Okay, so cara yang kedua sama. Ini adalah opsonization, kan, ini cara yang kedua terbahagi kepada agglutination and also precipitation. Precipitation for dissolve antigen. Okay, agglutination untuk to clump the bacteria together, so dia tak boleh nak bergerak dah. So then akan datang macrophage and then engulf, okay, or other phagocytes. And then yang ketiga adalah activation of complement system. So so this is the plasma membrane of the bacteria, contohnya ada antigen, lepas tu antibody bind, okay, and then dia akan aktifkan complement protein, kan. So complement protein will be inserted into the plasma membrane of the bacteria to form pores, and then water and then ion enters into the foreign cell and causes the foreign cell to swell and then burst. Okay. Okay. So that is the explanation. So kita discard dulu. Kita tengok dia punya video lagi. Media 2.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Mission accomplished.
Okay, so that's the explanation of how antibody lead to the pathogen being destroyed. Ingat ya, antibody will not destroy the pathogen directly. Dia akan lead kepada pathogen to be destroyed by neutralization, agglutination, precipitation, and also activation of complement system. Okay, so next, saya lupa nak present. Okay, so done with the humoral immune response. So now we are going to look at the second, third line defense, which is the cell-mediated immune response. Okay, so for cell-mediated immunity, so macam saya dah cakap banyak kali tadi kan, so this involve infected cell, infected body cell. So infected cell displays the antigen fragment on its surface using class 1 MHC. Ingat class 1 sorry. Okay, tersedak. Ada orang ada ngumpat ke? Alright. So cell-mediated immunity. So infected cell displays antigen fragment on its surface using class 1 MHC molecule. Okay. So an activated cytotoxic T cell recognizes and binds to the antigen MHC complex via TCR. TCR adalah apa? T cell receptor. Siapa punya T cell receptor? Cytotoxic T cell receptor. Okay. Cytotoxic T cell receptor of the cytotoxic T cell kan. So cytotoxic T cell dekat cell surface dia ada receptor. So receptor ni akan bind kepada antigen molecule that is presented by infected cell using class 1 MHC. Okay. So the activated cytotoxic T cell releases perforin. Okay, so ini molekul yang pertama lah, perforin. Okay. Perforin. So perforin that forms pores. So nama pun perforin kan. So it produces pores on the cell membrane of the infected cell. Okay. So the activated cytotoxic T cell then secrete a second type of molecule, which is granzyme. Okay, so this granzyme enters the cell through pores that induces apoptosis. So molekul kedua yang akan diriliskan oleh cytotoxic T cell adalah granzyme. So granzyme ni masuk melalui pores yang terhasil tadi menggunakan perforin. Satu cara. Kalau baca dalam buku Campbell, cara lain untuk granzyme masuk adalah melalui endocytosis. Itu dalam buku Campbell lah dia mention. Okay, so the function of this granzyme is to cause apoptosis to occur. So what is apoptosis? Is programmed cell death. It causes or cell suicide. Okay, it causes the infected cell to be destroyed. Okay, so programmed cell death ataupun cell suicide. So the killing action of the cytotoxic T cell on infected host cell. So this is infected. Okay, so this is infected cell. So this is the infected cell, an infected cell. So as we can see inside the infected cell, it has the pathogen inside it that has been fragmentized. Okay, so these fragments of the pathogen, okay, will bind to the class 1 MHC molecule yang ni terhasil oleh the infected cell tu. So we call it as antigen MHC complex. So akan datang nanti cytotoxic T cell with receptor that complements with the antigen being presented, okay, by the infected cell. So kalau tengok kat sini, so this is cytotoxic T cell. Inside it has two chemical. The first one is perforin, the second one is granzyme. Tengok kat sini, ada satu molecule, we call it protein, we call it as CD8. Okay, CD8 ni adalah sejenis protein yang dia adalah kepunyaan cytotoxic T cell. Okay, CD8 ni adalah molekul kepunyaan cytotoxic T cell yang mana kegunaan dia adalah untuk enhance the interaction between the cytotoxic T cell and infected antigen. So once you have this interaction, the binding of the receptor of cytotoxic T cell with the antigen molecule, okay, presented by the infected cell using class 1 MHC. So lepas tu akan ada interaction menggunakan CD8 ni. CD8 is a protein molecule from the cytotoxic T cell, which is the function of it is to enhance the interaction between the cytotoxic T cell and peptide antigen. So once you have this interaction, it causes the cytotoxic T cell to release perforin. Okay, it forms pores. So next, the cytotoxic T cell secrete yang granzyme ni pula, granzyme yang warna purple ni. So granzyme enters through the pores ataupun melalui yang ini perforin yang purple ni, yang bintik-bintik yang lebih kecil tu adalah granzyme. So the granzyme enters into the infected cell through the pores or through endocytosis. So next dia akan cause apoptosis, okay, ptosis, which is programmed cell death. So this causes the cytotoxic T cell to be destroyed and expose the pathogen. Okay. So another video we are going to play, and show this card. Sekejap. So media 3 will be playing in a moment. Kejap je.
Now let's look in more detail at cell-mediated immunity. Cytotoxic T cells are the main warriors of cell-mediated immunity, which attacks body cells that have been altered by infection or cancer, or transplanted cells that are foreign to the body. An infected, cancerous, or transplanted cell displays foreign antigens on its surface. T cell receptors on specific cytotoxic T cells recognize and bind to specific antigens on cells. Cytotoxic T cells respond to this contact and to interleukin-2 sent out by helper T cells by producing the protein perforin. Perforin molecules are released from the cytotoxic T cell and attack the membrane of the infected cell, punching holes in it, which allows water and solutes to enter. The cell bursts and is killed. In this way, cytotoxic T cells deprive pathogens of their host cells so they cannot reproduce. Tumor cells are also removed from the body by cytotoxic T cells. In this way, cell-mediated immunity, carried out by cytotoxic T cells, destroys altered or infected body cells.
Okay, so that's a video to explain on the mechanism of how cytotoxic T cell destroy infected body cells. Okay, so in the cell-mediated immune response. Okay, so now kita akan proceed to the next one. Okay. Kita rehat 2 minit sekejap ya. Sekejap je. Okay, so next, using this diagram, okay, it shows you, this diagram is from Campbell. So it illustrates, okay, how antibody-mediated immunity and also cell-mediated immunity works. Okay, so if you look at this diagram here, sama je, cerita yang sama je. Okay, so this is antigen. Okay, so kalau tengok antigen ni, this is for the first time exposure. Primary immune response. Note kat sini, primary immune response. Okay, so this when your body is first time infected by a certain virus or pathogen, it enters into your body, it circulates in your body fluid, it infected your body cell. Okay, so dia melibatkan antibody mediated immunity and also cell mediated immunity. So the antigen will be engulfed by the white blood cells that can be macrophage, dendritic and also B cell. Note kat sini ada 3 jenis, macrophage, dendritic and also B cell. So once the antigen is being engulfed by that 3 different cell, okay, either of the 3 different cell, it becomes APC, antigen presenting cell. So kalau plus sign ni adalah stimulate lah ataupun activate. Okay, so the APC present the antigen using class 2, okay, on the cell surface. So dia akan present kepada siapa? Kepada helper T cell. So once helper T cell with the receptor that binds to the antigen being presented by APC, so this causes the APC to release cytokine, okay, cytokine. So this causes the helper T cell to release its own cytokine. So this the cytokine released by the helper T cell activate itself, okay, and causes the helper T cell to proliferate and then differentiate into two colonies, which is one the effector cell, and the other one is memory. Okay, memory helper T cell. So memory helper T cell will be stored in your lymph nodes, for example, for future infection. Okay, future infection by the same virus or pathogen. Okay, so this is secondary immune response. Tulis kat sini, secondary immune response. Okay. So kalau tengok kat sini, so there's the cytokine which is released by the helper T cell also will activate cytotoxic T cell and also B cell. Okay, cytotoxic T cell in the cell-mediated immune response and the B cell for the antibody-mediated immune response. Okay. So bila kedua-dua sel ni dah diaktifkan, awak tengok so dia akan undergo, tulis kat sini proliferation, kan, proliferate. It divides into many cells, okay, many B cell with the same receptor, many cytotoxic T cell with the same receptor. So and then they akan undergo clonal selection. So clonal selection produces the effector for B cell which is plasma cell and also memory B cell that will be stored in the lymph nodes for future infection by the same pathogen. So kalau tengok kat sini plasma cell, the function of the plasma cell is to secrete antibodies that will attack the antigen, okay, the same antigen, that specific antigen. Okay, so kalau tengok kat sini, so kalau tengok kat sini, for antibody-mediated immunity, it involves, okay, to defend your body against extracellular pathogen. What does it mean? Extracellular pathogen? Pathogen that are outside of your body cell. Outside kat mana? Outside which is in your body fluid. Kan? Extracellular pathogen. Pathogen that are found in your body fluid. Blood, lymph, interstitial fluid. And as for the cell-mediated immunity, again, tengok kat sini, once the cytotoxic T cells proliferate and then undergo clonal selection to produce the active cytotoxic T cell ataupun effector cell dia akan juga menghasilkan memory T cell, memory cytotoxic T cell that will be stored in the lymph nodes for future infection by the same pathogen. So kalau awak tengok kat sini, for the cell-mediated immunity, it is to defend against intracellular pathogens and also cancerous cell. Intracellular pathogen are pathogen that are inside the host cell, okay, inside of the host cell. And also is to destroy cancerous cell. Okay, so kalau awak tengok sini, so this diagram basically it summarizes the whole story yang saya explain tadi tu. Kalau macam kita tengok kat sini kan, plus sign is to stimulate, right? Kalau tengok kat sini, mana mouse saya ni hilang. Okay. Kalau tengok kat sini kan, so antigen ni kan, so dia akan juga bind kepada B cell kan. So B cell ni akan boleh jadi APC juga, ataupun in order to activate the B cell, it requires cytokines, okay, which is released by the helper T cell. APC, okay, juga boleh terus aktifkan cytotoxic T cell. Dia boleh juga aktifkan cytotoxic T cell. Okay. Okay, so hemostatic imbalances in the immune system. So now we are going to look at imbalances in the immune system. Okay, so homeostatic imbalances is disruption. Okay, disruption in the immune system function occurs when the immune system responds to self-antigen, causing serious and some fatal diseases. So it can be divided into immunodeficiency diseases, autoimmune diseases, and also allergies. Kalau immunodeficiency adalah keadaan yang mana your immune system does not work properly. Your body, your immune system cannot protect your body from being infected by infection of pathogen or virus. Kalau autoimmune disease lebih kepada explanation yang ni. Okay? Yang kedua ni. Your immune cell, your immune response attack self-antigen. It attacks your own body cell, which is supposed, it cannot occur like that. Okay, so this one, it leads to autoimmune disease. Kalau allergies adalah keadaan yang mana when you are exposed to some allergen, it causes a reaction to occur. Okay, reaction, your immune response to react against that allergen. So allergen ni selalunya adalah molekul yang selalunya sepatutnya tak berbahaya pun, macam contohnya seafood, some chemicals from cosmetic for example. Okay. So certain pollen contohnya kan. So ada orang yang tak ada apa-apa reaction terhadap molekul-molekul ni, tapi ada orang yang akan boleh trigger allergic reaction. So that is allergic lah, allergens. Allergen that causes allergic reaction, which involve basically selalunya kalau allergic reaction ni dia melibatkan antibodies. Ya, antibody punya reaction kat situ. Nanti kita tengok the mechanism. Okay. So first kita tengok immunodeficiency disease. So it is a disorder in which the immune system is lacking. Okay, it is lacking. It cannot protect your body. Okay, it's lacking the ability to protect against pathogen. Highlight lacking tu sebab immunodeficiency, ketakbolehan immune system awak nak protect awak punya badan against pathogen. So leads to frequent and recurrent infection and susceptible to cancer. So maksudnya kalau orang ada immunodeficiency, so dia senang kena sakit lah sebab the body of that person is not protected by the immune system. Okay. So immunodeficiency can be divided into two, which is inborn immunodeficiency, which is congenital, congenital, and then acquired immunodeficiency, which is you get later in your life. Kalau inborn immunodeficiency, dia adalah disebabkan oleh macam contohnya genetik. Okay, so result from genetic or developmental defect in the production of immune system cells or specific proteins. Okay, maksudnya daripada baby lagi memang awak dah ada penyakit ni. Okay, you inherit that disease for example. Okay, so for example severe combined immunodeficiency disease SCID, which is shortage or absence of B and T cell. So imagine lah kalau there is no B cell, there is no T cell, how can your body be protected, okay, from specific virus? Okay, so B cell is to produce antibody, so your body cannot produce antibody. Okay, contohnya, ataupun kurang lah, shortage. Okay, so the person is susceptible to pneumonia and meningitis that can be fatal, can be treated by bone marrow and stem cell transplantation. Okay, so bone marrow transplantation because your blood cell to be produced again lah, can white blood cells that can be produced. So the second type is acquired immunodeficiency, or it develops later in life. So due to exposure to certain chemicals or biological agents such as cancer, drugs to fight autoimmune diseases, or drugs to prevent transplant rejection. So kalau macam contohnya orang yang ada buat transplant transplant organ kan, so that organ is considered as foreign to your body. Okay, so dia akan badan awak ni akan ada tendency untuk react against that organ. So for people, for your body, to prevent your body from reacting against that organ, okay, so you are given drugs, okay, so that this drug cause your body to not attack that organ. Okay, contohnya lah, okay. So kalau macam autoimmune disease kan, autoimmune disease maksudnya antibody dalam badan awak ni attack self antigen. Okay, so you take medication in order to prevent your antibody from attacking your own body cell, self antigen. Tapi instead virus drugs ni dia efek terlebih-lebih pula. Macam contohnya dia akan terus menyebabkan autoimmune disease. Tak boleh nak bertindak balas langsung kat situ. Okay. Acquired. Macam itulah. Okay. So example human. Okay. Human immunodeficiency syndrome. AIDS. Okay. AIDS. Acquired. Okay, so AIDS, okay, is caused by this virus, which is human immunodeficiency virus, which attacks helper T cell. So imagine kalau helper T cell is destroyed, okay, by this virus. So maksudnya tak boleh nak aktifkan B cell, tak boleh nak aktifkan cytotoxic T cell. So humoral immune response will not be activated. Cytotoxic T cell will not be activated. Kalau B cell tak diaktifkan, tak adalah antibody kan. Tak ada antibody, so your body will not be protected if the helper T cell is destroyed by the HIV. Okay, the human immunodeficiency virus. So virus dia adalah HIV, penyakit dia adalah AIDS. Okay, so the number of helper T cell will decrease, leading to a decrease in the number of B cell and also cytotoxic T cell. So the person is susceptible to infection and also cancer. Okay. So ini adalah immunodeficiency. Highlight kat situ, deficiency, tak boleh, lacking. Okay, your body is lacking immune response to protect yourself against infection by pathogen. Next one is autoimmune disease. It is a disorder in which the immune system fights the body own self-antigen, causing inflammation and organ damage. 2 out of 3 of the people with this disease are women. So women have higher tendency to get this disease. Okay, so there are several different types of autoimmune disease that we are going to look at later. So the autoimmune disease is also influenced by gender. Tadi kan women lebih ada higher tendency to get the disease. Also genetics and also environment. Okay, so affects nerves, muscle, endocrine system and also digestive system. Well, for example, we have rheumatoid arthritis. Rheumatoid arthritis is the condition where if I'm not mistaken, is antibody that attacks joints. Okay, it attacks joints yang dekat tangan, dekat jari-jari tu. It attacks joints dekat lutut, joints lah dekat kaki kan. So kalau tengok gambar, kalau orang yang ada rheumatoid arthritis, if not treated, dia akan menyebabkan jari tu bengkok dan it is really painful. Okay, it is really painful. Okay, so this one, rheumatoid arthritis, it attacks joints. Inilah okay, rheumatoid arthritis. So a disease in which the cartilage and joints are damaged and inflamed. Okay, causes deformed joints accompanied with muscle pain, weaknesses, fatigue, loss of appetite and also weight loss. Okay, so kalau orang yang severe sangat, it could lead to this deformed joints ni. Okay, kalau selalunya autoimmune disease ni dia tak boleh sembuh, tapi ada medication untuk kurangkan kesan dia, macam tu. Okay, so allergies. Okay, so allergies are hypersensitivities responses to allergens. Okay, allergens or antigens. Okay, so allergen consists of various foods and drugs, insect venoms, molds, fungus spores, dust mites, cosmetics and also pollen grains. Okay, so occurs in the nasal passage, bronchi and also skin. For example, you have hay fever. So allergic ni dia akan menyebabkan if you are exposed to the allergen, it could cause you to sneeze, for example, itchiness at the skin, for example, macam tu kan. So kat sini dia cakap occurs rapidly in response to the binding of antibodies to a tiny amount of allergen. So in the first statement here, dia nak cakap, okay, when you are first exposed to the allergen, okay, when you are first exposed to the allergen, it causes certain B cell to be activated and produce the antibody. Okay, so it says antibodies. So antibody yang terhasil ni adalah antibody jenis IgE. Antibody ni kita ada beberapa jenis, ada IgE, IgG, there are IgA kan. So there are several different antibodies kan, yang ada different peranan, tapi in this case in the allergic reaction it involves IgE. Okay, so excessive amount of IgE is produced in response to the initial exposure to the antigen, okay, or to the allergens. So the IgE binds to mast cell. Kita dah tengok hari tu kan, inflammatory immune response. In the mast cell, okay, mast cell mast cell tu sel yang ada pada your body tissue. Okay. So mast cell ni dalam dia akan ada vesicles. In this vesicle it contains histamine. Okay. So dekat cell surface of the mast cell akan ada receptor. Okay. So the receptor of the receptor on the mast cell will bind to antibody. It will bind to antibody macam ni. Will bind to antibody. Okay. So allergen ni dia akan bind antara dua antibody yang bind kepada mast cell punya receptor. Yang ini kita panggil interaction ni sebagai cross linking. Macam itulah. Okay, cross linking. So when you have this interaction, it causes the histamine inside the mast cell to be released. Okay, histamine. Okay, histamine. So histamine ni kita belajar hari tu kan dalam inflammatory response, it causes vasodilation of blood vessels, more blood to flow to that area. Macam tu lah kan. So when this occurs, it causes certain areas such as your nose, kan, your bronchi, nasal passage to be more sensitive. Okay. So upon subsequent exposure to the same antigen, the antigen cross-link, inilah istilah dia cross-link ni kan, to neighboring IgE, and this triggers the mast cell to release histamine. So the release of histamine causes increased vasodilation and capillary permeability. Okay, so symptom result in mucosal edema, redness, can, so because more blood flows to that area, rashes, runny nose and sneezing. Okay, so can be treated using antihistamine and also epinephrine. Okay. Okay, so kalau tengok kat gambar ni okay, so initial yang awak kena, gambar yang ni awak kena tengok start daripada nombor 1 ni. Okay. Okay, so ini kita ada allergen. Contohnya pollen lah. Okay, so pollen. So pollen binds to the receptor on B cell. Okay, it binds to receptor on B cell. So B cell ni akan macam tadi kan, dia akan undergo clonus, it will be activated and then it undergo proliferation and also clonal selection. Okay, so clonal selection akan hasilkan plasma cell. So plasma cell akan secrete lots of antibody, okay, with the antigen binding site that matches with the allergen tu tadi. But the antibody release, we call it as the IgE. Okay, IgE. So this IgE molecule, antibody ni, akan bind kepada receptor on the cell surface of mast cell. Okay, so when you have subsequent exposure to the same allergen, kan, ini subsequent ni first exposure, subsequent exposure. Okay, so when you are exposed again to the same allergen, okay, dia akan trigger allergic reaction dengan cara macam mana allergen ni akan bind between two neighboring IgE antibody on the surface of the mast cell. So dia akan bind, dia akan menyebabkan interaction ni kita panggil dia sebagai cross-link. Okay, cross-linking neighboring IgE on the mast cell. And this can lead to, we call it degranulation. Okay, degranulation, which means the vesicle inside the mast cell that contain histamine will be released. The histamine will be released outside and causes tadi vasodilation lah semua ni kan. Vasodilation, increase capillary permeability, so that leads to all these symptoms. Okay, mucosal edema, redness, rashes, runny nose and also sneezing ni. Okay. So ini adalah mechanism for allergic reaction. Kalau ni nampak the subsequent contact with the allergen. Macam tu lah. So macam contohnya kalau ada orang tu dia initially dia boleh makan seafood. Okay, tapi lama-lama dia boleh develop allergic. Sebab ni lah interaction macam ni kan. Ada antibody that was produced against the allergen that has bind to the mast cell. So mast cell akan release histamine, okay, due to cross-linking of the allergen. Okay, so kalau tengok kat sini sama je cerita dia, initial and subsequent exposure. Okay, so initial exposure. Okay, ini initial exposure. So you have the mast cell. okay, that was initially binds to the antibody that was produced and secreted by plasma cell kan. Plasma cell. So plasma cell yang sebelum tu akan ada B cell yang bind kepada allergen tu tadi. Okay. So plasma cell secretes antibody. Antibody binds to the receptor on the mast cell. When you are exposed again, okay, exposed again to that same allergen, okay, so dia akan bind kepada neighboring antibody on the mast cell. So this causes degranulation. Okay, degranulation, which is the release of histamine from the mast cell that could lead to all the allergic symptoms. Okay. So next is the last part is infectious diseases. Okay. So infectious diseases is what? Caused by pathogenic microorganisms such as bacteria, viruses, parasites, and also fungi. Okay, can be infected through direct contact with the person that is sick. Macam contoh kita sekarang ni lah situation COVID ni kan. So the person that has the COVID has to be quarantined so that you are not exposed to the virus. Okay. So indirect contact when you touch something that has germs on it. So sebab itulah kena ada berlaku sanitization kat situ kan. Bawa pergi mana-mana kena bawa sanitizer macam tu kan. So insect factors such as Anopheles mosquito. Okay, so yang ini dia bawa pembawa lah kan. So yang bawa virus. Okay. So ataupun melalui contaminated food. Okay, so makanan dah tercemar, tak boleh makan dah. And also water, sebab tu kena masak air, soil and also plants. Okay, so apa-apa, kalau siapa yang suka buah ke sayur ke whatever, make sure that you wash it first. Okay, jangan telan je. Pergi kat pasar tu kan, buah-buah yang dijual tu dah berapa orang dah pegang. So you make sure you wash it first. Okay, so first is dengue. So viruses are transmitted through the bite of infected Aedes aegypti and Aedes albopictus, okay, which is female mosquito during daytime. Okay, so should be suspected when a high fever or demam yang suhu dia tinggi, which is 40 degrees Celsius, which is accompanied by these symptoms: severe headache, pain behind the eyes, muscle and joint pains, nausea, vomiting, swollen glands and also rashes. Okay, so dengue ni masalah kat tempat yang selalunya considered macam tempat yang tu bersih. Tapi bersih sebab apa? Contohnya dalam perumahan yang bersih-bersih ni, ada yang suka tanam pokok kat pasu tapi dia tak clearkan air yang bertakung contohnya kan. So sebab tu kalau dekat bandar-bandar banyak berlaku dengue ni kan. So due to all this keadaan kan, so ada pasu yang bertakung, tayar semua. So this could lead to the breeding of the mosquito punya istilah dia jentik-jentik tu lah. Okay, alright. So prevention. So wear natural colouring clothing, long sleeve and breathable garment, removing container and disposing of unwanted rubbish around the yard that may collect water. So apa-apa yang awak rasa boleh contain ataupun store water, you have to remove. Okay, walaupun cantik pasu bunga awak tu, make sure tak ada yang bertakung. So store containers upside down. Let pots drain directly onto the ground, avoiding using saucers or dripping trays under pot plant. So ada yang letak kat bawah tu kan, dia punya saucers dia tu, dripping trays. So elakkan guna ni, terus letak pasu tu atas rumput ke atas tanah ke kan. So use of personal household protection, window screen, mosquito nets, long sleeve clothes, insecticide treated material, coils and vapors, surface spray, okay, and fumigation. So vaccine, people living in some dengue endemic country. So ada yang macam kawasan yang memang dah teruk, negara yang teruk tu ada vaksin. Okay. Next is TB, tuberculosis. Okay. TB is caused by bacteria Mycobacterium tuberculosis. Okay, that most often affect the lungs. Okay, tuberculosis is curable and preventable. Okay, so kalau batuk-batuk kering semua tu, batuk je, please avoid coughing directly. Okay, to other person. Kan, itu etiket lah kan. Okay, so prevention. So one is immunization. Ada vaccine against TB. Kan, TB vaccine, BCG is often given to babies and also children. Improving the ventilation in indoor spaces, fewer bacteria in the air. Kan, so make sure ada aliran udara yang baik lah kan kat situ. And then using germicidal UV lamps to kill airborne bacteria in buildings. Okay. So treating latent infection before it becomes active. So infected people has to practice cough etiquette, okay, and respiratory hygiene. So kalau dah batuk tu, tutuplah mulut kan dengan kalau boleh dengan kain ke dengan tisu kan. So and then respiratory hygiene. So tangan yang dah kena bersin tu tadi, batuk ke whatever, make sure you wash. Kan, jangan dah batuk, dah bersin semua, pegang merata-rata. Okay. And then the next one is hepatitis. So hepatitis is an inflammation of the liver. So in chronic hepatitis, liver inflammation continues for at least 6 months. Okay, so this condition may be mild and result in relatively little damage, or may be more serious, resulting, okay, in destruction of many liver cells. Okay, because destruction of many liver cells. So severe cases. Some severe cases leads to cirrhosis and eventually liver failure. Okay, so as you can see from this, kita ada hepatitis A and also hepatitis B. So hepatitis is a risk for travelers worldwide. Okay, so over 1/4 of the world's population have been infected with the hepatitis B virus. Okay. So it says here 1.4 million causes of hepatitis A are identified worldwide each year. So the number of passengers that feel so maksudnya boleh muat sampai 4,000 Boeing passenger ni kan. So berapa ramai orang yang orang kena hepatitis ni, 1.4 million worldwide ni lah. So hepatitis A can cause acute liver failure, which is associated with high mortality. So maksudnya boleh menyebabkan kematian lah, high mortality ni. Okay. So according to 2014 WHO's analysis, investment in hepatitis B vaccine could prevent an estimated 4.8 million death, okay, over 10 years period. Itu untuk hepatitis B lah. 10th leading cause of death worldwide. So kalau tengok dekat dalam map ni, prevalence of hepatitis A, tengok kat sini, contohnya dekat dia basically paling banyak dekat third world country lah ni. Okay, dekat India, dekat South Africa. Prevalence of hepatitis B dekat area-area merah tu. Okay, so daripada data. Okay, so hepatitis A, the transmission is through fecal or oral. Okay, so common in areas for poor sanitary condition. Epidemic can occur in situation of water contamination. So kalau kita kat sini kat negara-negara yang tak berapa nak bersih lah kan, tak berapa nak bersih. So kalau kita tengok kat sini macam dekat India, kat Afrika sini kan, so tak berapa nak bersih dia punya keadaan hygiene tu. Okay. So kalau hepatitis B, transmission occurs via body fluid, blood, semen, etc. So high risk group include medical personnel, male homosexuals, and also dialysis patient. So hepatitis C, the virus is usually spread by direct contact with the blood of an infected person, including sharing needles and also blood transfusion. Okay. So prevention, ada vaksin. Okay, so against hepatitis A and B. So you always have to make sure you wash your hand thoroughly after using the restroom, and when you come in contact with an infected person's blood, stool and other body fluid. So pergi ke tandas-tandas awam ni, make sure it is clean. Okay, make sure it is clean. So avoid unclean food and water. Can be infected by eating food or drinking water that has the hepatitis A virus in it. So avoid contact with other people's blood and body fluid. Don't share toothbrush, okay, razors and also nail clippers. Okay. So ini adalah jadual vaccination, okay, jadual vaccination yang ambil daripada baby sampai remaja. Kan? So kalau siapa yang still ada adik-adik baby lagi, kalau ada buku baby tu, so dia ada jadual vaksin tu kan, daripada baby to 1 month to 2 month, 3 month, 4 month, 6 month. So ini mesti kena pergilah follow up untuk vaksin. Okay, kalau kita tengok kat sini, kalau so dia ada dah siap-siap untuk hepatitis B kan. Ada untuk diphtheria, ada untuk polio, measles, okay, MMR. Macam itulah. Kalau untuk yang perempuan ada HPV kan. And then kalau tetanus booster. Tetanus booster ni kalau siapa yang mengandung, dia akan kena cocok lagi. Siapa yang mengandung sebelum dia dah bersalin, dia kena cocok tetanus lagi setiap kali. Okay, mengandung nak bersalin tu, so akan cocok tetanus. Okay, so how about COVID-19? So let us discuss about this disease. So samalah apply kepada COVID-19. So virus ni dia boleh infect dengan easy lah if you do not take care of your SOP. Macam tu kan, kena selalu jaga SOP, bawa sanitizer, bawa pakai face mask, semua tu kena jaga jarak and all that. Okay, so this is to prevent the spread of the virus. Okay, so that's the end of chapter 7. Okay, so do you have any question?